Working memory is a crucial part of human functioning that helps us to carry out activities, such as recalling a recipe for your favorite meal. Working memory allows us to temporarily store and manage information that is required to complete complex cognitive tasks like learning, reasoning, and comprehension, and is involved in the selection, initiation, and termination of information processing, such as encoding, storing, and retrieving data. A large contributor to the working memory system is the visual system, enough so that it has its own name, the visual working memory system. Visual working memory provides an essential link between perception of visual stimuli and its corresponding processing and higher cognitive function, which allows for the active maintenance of information of stimuli when it's no longer in view. In the visual system, there are a series of synapses that transmit visual information to the brain for processing. In regards to the visual working memory system, there are several brain regions that activate during visual information processing. Previous research suggests that higher-order processing areas, which include the prefrontal, parietal, inferotemporal, and lateral occipital areas, support visual maintenance and account for the limited capacity of approximately 3 to 4 items in the visual working memory space. There's a lack of research surrounding these early visual areas and their contribution to visual selectivity. It has been shown that higher-order areas lack visual selectivity of early sensory areas, so it is unclear how people can remember specific visual features with minimal decay over seconds. One theory suggests sensory areas serve to maintain fine-tuned feature information, but this cannot be true as early visual areas show little to no sustained activity over prolonged delays. So, how are early visual areas involved with the visual working memory system?
Harrison and Tong addressed this question and hypothesized that early visual areas can retain specific information about visual features held in working memory over periods of many seconds when no physical stimulus is present. To validate this, Harrison and Tong outlined several studies. First, they sought to investigate the role of early visual areas in working memory. They hypothesized that pattern classification methods can successfully recover orientation information from cortical activities sampled at resolutions using fMRI. Second, they investigated whether activity patterns during a delay period would predict which of two orientations was held in working memory. They hypothesized that maintaining an orientation is associated with widespread changes in orientation selective activity throughout the early visual system. Lastly, they investigated whether an orientation selective activity is maintained throughout the working memory delay period. They hypothesized that the orientation selective activity persisted throughout the period when no physical stimulus is present.
In terms of materials, researchers used a 3 Tesla Philips Intera Achieva MRI scanner and used a gradient echo echoplanar 2-star weighted imaging system for functional images. They had 6 participants between the ages of 24 and 36 with normal or corrected to normal vision. In this paradigm, the researchers wanted to look at memory-specific activity. So they had 2 samples, 2 near orthogonal gratings at 25 degrees and the other at 115 that were presented in a randomized order with a fixation point in between. The participants were then given a number cue, either 1 or 2, which indicates which sample they should remember. It is important that this cue was given after the gratings were shown and not before, because the subjects would pay more attention to the grating that they were cued to remember. Then they were given an 11-second retention period, and then a novel test grating was presented, and then the subjects had to report whether the test was rotated clockwise or anti-clockwise relative to the grating that they were told to remember.
Okay, so in terms of the results for this task, participants were able to discriminate small differences in the orientation of the gratings between the cued and the test ones, and their performance was equally as good when it was either the first or the second grating to be remembered. In particular, there was a 75% remembrance rate for the first one and 73% for the second, which is not a significant difference.
In order to see if activity in early visual areas would give us any information about the contents of working memory, researchers used an fMRI decoding method. They were looking at if activity patterns during the 11-second retention period might predict which of the 2 orientation gratings was being held in the subject's working memory. Researchers therefore calculated an average response of individual voxels over time points 6 to 10 seconds in early visual areas, and this was to account for the bold response hemodynamic lag. Then they used these values from each trial as an input to create a linear classifier. And finally, the classification accuracies were determined using cross-validation methods.
So as a result of the fMRI decoding, it was seen through the green curve that activity in areas B1 to B4 end up being a good predictor of orientation that was being held in working memory. The classification accuracies reached about 83%. So individual areas themselves also showed similar levels of decoding performance. So this tells us that maintaining an orientation in working memory can be associated with changes in orientation selective activity in the early visual system.
So next, the researchers wanted to see how orientation selectivity response for remembered gradings would compare with stimulus-driven activity, which would technically be elicited by direct viewing of actual gradings. So the researchers did a new experiment now where participants would report whenever a J or a K appeared in a sequence of centrally presented letters. There were about 4 letters per second. And in the meantime, task-irrelevant gradings flashed on and off in the background every 250 milliseconds. And these gradings were the exact same as the ones used in the first experiment with the working memory. They were just at a lower contrast. So they found that even though these gradings were faint and unrelated to the task, the gradings still evoked orientation selective responses in early visual areas, as you can see in the red curve over here. However, as you can see in this graph, performance was quite lower for the individual areas V3A and V4, and this could be because activity in higher extra striate areas, it may actually be more dependent on visual attention. And the gradients in the background, in this case, were not being given as much attention, which is why in the first experiment, in the green line, it might be of higher values. In order to compare both experiments of working memory and stimulus-driven activity and their similarity in terms of their orientation selective patterns, researchers trained a classifier, where they would use one dataset and test it on the other. And the comparison and classification accuracies of these two experiments tells us that retaining an orientation in your working memory may recruit many of the same orientation selective subpopulations of neurons as those that are activated under stimulus-driven conditions, due to how similar they were.
Finally, researchers wanted to see whether orientation selective activity is maintained throughout the working memory delay period by using decoding analysis on specific individual fMRI time points. This would allow us to detect changes in orientation selective activity over time in the 2 previous experiments. We can see that the working memory response lagged by about 2 seconds behind the unattended gradings task, and this may be due to the time that was needed to interpret the cue that was given, telling the participants which grading needed to be remembered. But the main thing that we see here is that orientation selective activity persisted throughout the 11-second delay/retention period, even when no physical stimulus was present.
The results from this experiment showcase multiple interesting implications regarding the early visual system that challenge previous literature on the subject. Before now, our understanding of the visual system was that early visual areas could not retain specific information about visual features in working memory. In Harrison and Tong's experiment, as we just discussed, when participants had to remember a precise orientation, specifically the cued grading orientation in the experiment paradigm, fMRI decoding methods demonstrated that orientation-specific activity patterns were retained in primary visual areas. Also, the study outlined possible increasing evidence that suggests higher-level support functions for Area V1, an early visual area which we've discussed in class, such as conscious perception, attention selection, and other complex cognitive functions, in addition to its essential low-level feature processing. In the working memory task, the study also presents results which may indicate that early visual areas can maintain information in the absence of direct input to stimuli. This demonstrates both the sensory processing function of these neurons, as the literature has already established, as well as the new possible function in the maintenance of visual features and patterns in memory. There has also been little evidence in the literature to suggest a link between V1 activity and visual working memory. This study found that while overall activity in the visual cortex fell to near baseline levels after prolonged delay, the decoding of low-amplitude signals could lead to a reliable prediction of orientation in visual working memory. This suggests that there is an important source of memory information that has been thus far unexplored, potentially because previous studies have been focusing on single neuron studies, which may have overlooked visual memory performance due to correlated noise, whereas population decoding methods such as these may lead to greater success moving forward. Some limitations of the study include the number of participants. The study only had an N of 6, and the power achieved was not indicated in the analysis. Another limitation indicated by the authors is that the decoding of remembered orientations could be mediated by eye movement. They explain that this may be unlikely due to the presentation of the stimulus being too brief for an associated eye movement, but this may need to be considered nonetheless. Further steps for related studies may look at the long-range interactions between higher-order areas and early visual areas. We know that current network models of working memory emphasize the importance of local recurrent activity, and these orientation-selective neurons in the visual cortex outlined in the study may be the necessary infrastructure for this local recurrent activity.